Morning, everybody. Um, I'm very delighted to start off today's exciting sessions with the first session. I'm Heather Jaspan. I'm here at University of Cape Town and at University of Washington and Seattle Children's Research Institute. I am going to recap a little bit of yesterday and also go over a few new points that I wanted to make before we start the session. Which is entitled The Ecology of Reproductive Wellness. Okay. So I think we got the point yesterday that women are the center of life, but we experience many changes throughout our lives, both socially and hormonally and health-wise. Starting from childhood to pre-sexual debut, then our reproductive, sexually active years. Women in Africa, for example, spend almost a quarter of their lives either pregnant or breastfeeding. So that's a huge portion of our lives. And then we have to undergo perimenopause and the postmenopausal period. And during these times, the vaginal microbiome plays a large role in our quality of life, in our health and the outcomes of our infants at the pregnancy periods and throughout our lives, throughout our sexually active lives. It plays a big role in risk for HIV and probably STIs, although some of that's not causally proven. And I just want to make the point that some of the data I'm showing, or some of the facts that we believe about the vagina and the vaginal ecosystem, are very much from the global north, because there's very little data from Africa. And here's a map that I sort of self-curated, showing you where there's data on vaginal microbiome in Africa. And most of this data is very just descriptive and structural, and not a lot of functional data has been published. For those who don't live and dream the vagina, I think I need to introduce that to be an optimal vaginal environment is when the vagina is dominated by a single lactobacillus species. So there's very low diversity in the microbiome, and this is accompanied by a low pH and a very intact mucosal surface. However, in dysbiosis or bacterial vaginosis, shown on the right here, these lactobacillus species are either replaced by or accompanied by a diverse set of facultative anaerobes, such as Gardnerella and Prevotella, and this causes inflammation. There's a high pH and there's barrier damage, probably because of the inflammation, but it's probably a multifactorial. And when this happens, obviously we're more susceptible to STIs, HIV target cells, for example, could be recruited. So this shows, again, from the global north, that women experience enormous fluctuations through their lifespan in estrogen and progesterone. And this is accompanied by decreases and increases, sort of inverse decreases and increases in the vaginal microbial diversity, which we call many things, but Shannon index is the one shown here. You can see that pre-adolescent girls have a very high vaginal microbiome diversity due to low hormone levels. This fluctuates a lot once they start to menstruate. As I mentioned, pregnancy is a high hormone level period of our lives, and that's when we have a low diversity in our vaginas, probably to protect the fetus. And then once we reach peri and postmenopause, when hormone levels drop, we again experience very high vaginal microbiome diversity. And This is again just to recap it a little bit more about the rest of the environment in the vagina. Pubertal girls have a very thin vaginal epithelium, low estrogen levels, low levels of lactobacillus, not necessarily BV-associated bacteria, but a high diversity. And then in the optimal state, once we have menarche, we have a thicker vaginal epithelium with lots of lactobacillus, low diversity, a low pH, as I mentioned, which is even more optimal during pregnancy. And then postpartum, again, we go to this thin epithelial barrier, low estrogen, high pH, and high microbial diversity. So for about a decade now, there's been quite a few descriptions in the global north that race and ethnicity may be associated with vaginal microbiome differences. So for example, this paper, from a very large paper shows pregnant and non-pregnant women on the top and bottom respectively, and women of African ancestry. This is US African American or US Black people. And you can see straight away that Lactobacillus crispatus takes up, or the women, there's very few women with Lactobacillus crispatus dominated vaginal microbiomes compared to those of European ancestry. But in more recent years, we've started to wonder if this is social determinant of health rather than race. So let's turn to Africa. And this is a map that I sort of curated myself and added some from these reviews that we've heard about the last couple of days. And as you can see, the map of Africa is quite well represented in gut microbiome research. But I showed you this map before. There's really a paucity of data in West Africa on vaginal microbiomes. Maybe Prof. Lebeer will tell us a little bit more today, and really practically nothing from North Africa, where speaking about vaginas is probably a little bit of taboo. So in addition to not having very well geographical description of the vaginal microbiome, we also have data that very much centers on women's sexual years and pregnancy. And there's a paucity, if not no data from Africa on pre-menarche, pre-sexual debut, and in the peri and postmenopausal time periods. Okay, here are some data from us, where we were lucky enough, or fortunate enough, I guess, to enroll very similar women from 3 settings in Africa. Women were 18 to 35 years of age. They were all at high risk for HIV, and they were all seeking effective contraception. So we know that they were sexually active. And I know it looks scary, but please bear with me. Each column is a woman. Each of the bottom rows are some of the most dominant taxa we found. And you can see here that women without BV, with a negative Nugent score, have predominantly Lactobacillus inners or Lactobacillus crispatus dominated vaginal microbiota and a low diversity. And women on the right-hand side have very highly diverse vaginal microbiomes shown here. They're BV positive by Nugent and have high Shannon entropy. But what I want you to focus on is this bar here, where we have a site shown, and the women from Cape Town are in orange versus the women from Kisumu, Kenya, and Johannesburg. And you can straight away see that the orange women, the Cape Town women are taking up a great portion of that diverse, more BV-associated vaginal microbiomes. And when we look at other data from South Africa, also our data, and here is a similar heat map, but supervised by site. We recruited adolescent girls, very, very similar HIV risk, similar setting. And here, here you can see on the left, we've supervised by site our adolescents from Cape Town or from Masiphumelele, if some of you went on Monday, and on the right from Soweto. And this is even more striking. These girls in the same country, the same sort of risk factors, But you can see that the girls from Cape Town, more than 50% of them have highly diverse BV positive vaginal microbiomes versus those from Johannesburg. So even within the same country, adolescent microbiomes differ. So I think I've made the point that we lack geographically distributed data from Africa, that we lack data from the extremes of age, and we really need more granularity in our vaginal microbiome data. And with that, I'd like to ask everybody to think through the next session and beyond, how can we accelerate more equitable vaginal microbiome research on the continent? And with that, I'd like to start the session. We have 3 fantastic female scientists, each of whom has already or will contribute substantially to African vaginal microbiome research. And I'm going to start with, well, Prof. Lebeer is going to start. She is a distinguished research professor in the Department of Bioscience and Engineering at the University of Antwerp, and she serves as the vice president of the International Scientific Association for Probiotics and Prebiotics. And she's going to tell us about a project that she spearheaded called the ISALA Citizen Science Project, and it's really transformed the understanding of vaginal lactobacilli in women's health. And this project has spread worldwide, and I'm very excited to hear about it.
Thank you very much for this kind introduction. Wait for my slides. Thank you very much for the great introduction also, Heather, because I want to summarize what we know about the vaginal microbiome. I'm a lactobacillus molecular biologist, is that it's dominated by lactobacilli in healthy women. At least this is based on most healthy cohorts studied so far. And also the associations that it's associated with health and reduced risk of infections, it's mainly based on epidemiological research, descriptive research anyway, but less on real molecular mechanisms of action. This is a map one of my postdocs made just based on the studies between July 21 and July 23, showing indeed again at the geographical distribution of vaginal microbiome research, where you see that Europe, although we have a bit higher percentage of studies carried out, this was before a Belgian study. I also must admit, after hearing yesterday all the different talks, that in Europe, also the research is very narrow and not centralized. And hearing now all the inspirational talks from Africa, I think Europe can also learn a lot from these collaborative initiatives. And then when you look just, again, similar, when you look at the studies that have been carried out, most are focused on infectious disease, on pregnancy, on diseases. And as a molecular fundamental scientist, I really think we lack just a fundamental understanding of the vaginal microbiome ecosystem. But we also lack the tools to properly study it. And a few years ago, as molecular biological scientists, we were really struggling with, we want to study lactobacilli in their proper niche in the human body, in the vagina, how to study lactobacilli in healthy women outside the clinical study setting. How can we access women? How can we ask women to donate samples for fundamental science studies, not for medical studies? So today I will indeed talk about our citizen science project. We have published a paper now almost 1 year ago, and it was also nicely covered in Nature Medicine. And it's a way we try to find a way to map the vaginal microbiome, but immediately link it also to functional insights and functional life course and lifestyle factors. So with the team, we were brainstorming how can we do studies in healthy women in an ethical, integrative way. How can we motivate them? How can we ask them to donate samples? And we were pitching it a bit also among friends. And then we soon realized if you want to do proper studies on the vaginal microbiome, you need to think properly about your sampling campaign, your communication. And one thing we soon decided is that we should first have a good communication campaign and good name for the project. So we decided for the Belgium project, we decided to name it the Isala project to honor Isala van Dis, who was the first female doctor in Belgium. And she seems like a very good role model. She was, for example, not allowed to study medicine in Belgium. She had to go to Switzerland. And if she came back to Belgium, she was not allowed to open a practice as a woman. So they had to change the law. And finally, she was allowed to open her practice. And from the beginning, she focused on vaginal health in a way, because she had, for example, a lot of prostitutes in Brussels, which she helped with their vaginal problems. And we found out that this naming the project Isala was also a way to facilitate communication about this kind of taboo topic, the vaginal microbiome. So from the beginning, I knew my scientific goals. I wanted to have a better understanding of the prevalence of lactobacilli. First of all, in Belgium, because in Belgium there were no vaginal microbiome research studies available, or not real detailed molecular studies. I want to understand the impact of lifestyle and life course factors to better understand when do lactobacilli occur. And also I wanted to set up a large biobank of isolates from the vagina to do research, but also to future work on innovative microbiome therapies. And we soon realized that this is our scientific goals. How does it matter for the women participating to your study? And we soon realized that if we are going to ask these very intimate samples to healthy women, we have to think about what can we give back to them, and societal goals, but also immediate impact goals for them. And we soon realized, for example, actually vaginal health is still a taboo topic. So that was the first goal we wanted to do, was break taboos on different aspects. During the past years, for example, we talked a lot about menopause, menstrual hygiene products, vaginal complaints, the Ramadan, or what has the impact of certain traditions on your vaginal health. The second thing was that we wanted to empower women, that they could take their own samples in the privacy of their own bathroom, that they can learn their own body, but also we wanted to embed it in a communication plan that we, together with the men and the partners and the babies, so that it was not only a female dominant project. And also the third goal was that we wanted to bring women in science and diversity into the picture. For example, the bioinformatics courses which we teach, it's still a bit male-dominated, and we also want to stimulate women that you can also big data analysis and some of these more technical expertise are very relevant for our field. Then I also wanted to pay attention to sampling box. So the project was not medical and not. So I wanted also to sampling box to look like almost like a gift box, so that women participating in our project would feel like properly acknowledged and would feel honored. And also we also paid attention to our website. We made a very nice website. We also made an underwear logo, which had a touch of humor, so that's also the communication was not always too scientific. And then we also have an Instagram page and Twitter now, or X, I must say, and a LinkedIn page. And we did all these efforts to recruit 200 women, because at that time we thought that we would be very ambitious if we could recruit 200 women. I must say we submitted a project to 5 times, I think, to our National Science Agency. They always said that you are way too ambitious with your 200 participants. So we never got a grant for the vaginal microbiome research. Then luckily I got an ERC grant on fundamental ecology and evolution of lactobacilli in different habitats and environments, also in fermented foods. The vagina was part of it. And finally, we had money that we could start. So we launched a call when the project started. And the enthusiasm was overwhelming. Within 10 days, we had 6,000 women registering on our website. So it was totally the opposite of what all grant agencies had told her. We had a lot of women did want to participate. Then I thought, yeah, maybe they don't realize what they have to do, because they have to give a lot of intimate data. They have to self-sample. But more than 4,600 women did fill in the very long questionnaire. It was a 1-hour questionnaire. They were willing to provide the data. And then they also were asked to send back, they received a sampling box, you can see at the top, they were allowed to send it back within 2 months, and then we stopped. And more than 3,300 women did do all this big effort. So it was really a huge, unexpected success. This is an example of the map of Belgium, which is a very small country in Europe. But as you can see, almost every little village in Belgium did participate. I had to speak Dutch, so we only have the Dutch-speaking parts of Belgium, but a little bit in the French-speaking parts. The age range, the minimum age was 18, and we had women up to 98 years old participating, so that was also very nice. And a bit of demographics, we still have 219 women around perimenopause and after and menopause, which is not the biggest part of our cohort, but still quite a nice number. And also we had actually quite some detailed data on contraceptive use, which also later we heard that the government, for example, or the policymakers were very interested in. So all the data you can find in our publication. Because the women were our most important stakeholders, we made sure that as soon as we had the samples, that we would sequence them all together and that we would give back the data to the participants. So they all received their personal profile. Here's a summary of our results. So most women, healthy women outside the hospital, were dominated by Lactobacillus status in our cohort, Lactobacillus inners, and then the more diverse communities. And that's actually also the first message we communicated to the general public is, good news, most women have a dominance of lactobacilli. And then we talked about the importance of lactobacilli. And then we also made communication channels on the website and also for healthcare professionals. We made infographics, but also we tried to be honest about what science already know. What do we know about the different bacteria? Where do they occur? And it was not difficult for lactobacilli, but it was more difficult for Prevotella, Gardnerella, the other microbes in these healthy women, because if we said honestly, to be honest, this is maybe not the best microbe to have in your vagina, but if you don't have symptoms, actually science doesn't know all the details and we need to investigate this further. And I must say that we spent quite some time on how shall we communicate this to healthy women. We should not alarm them, but we should also be honest. If you have symptoms, go to your medical professional. And it was actually nice to see that I told the team when we sent out the email to the 3,300 participants, be ready because probably we will get a lot of emails from worried participants. Actually, we had no email from a worried participant because they said we understood the message, but it's dynamic and what we had to do. But this feedback was actually very nice. Then, because we had so many samples, for the first time, we could really map the impact of different lifestyle and environmental factors in association with the vaginal microbiome. This is a summary of all the statistics, so I will not go into detail, but what we were able to do is rank different lifestyle and life course factors and link or associate them to different parameters of the vaginal microbiome, beta diversity, alpha diversity, the CSTs. We also made models or eigentaxa of co-occurrence patterns and then individual taxa. All these data can be found on the ANI, the European database, but also the metadata are publicly available. For this metadata, of course, they're very privacy sensitive, so you have to file a request and then it will be reviewed. So I don't have time to go into detail, but this is a summary we made for the general public about the factors for which we observed significant associations, age, natural course of your menstrual cycle, whether you have children or not, and that was a bit of a surprise to us, use of hormonal contraceptives, use of period products, foods, sexual activity, but we also made a message that it's a dynamic system. It's often stable, especially if you have lactobacilli, but not always. I will just zoom in on one aspect because we didn't plan it, but because it's a citizen science project, women said, you have to investigate associations with menstrual hygiene practices. To our surprise, we did find quite some significant associations. For example, wearing a menstrual pad was associated with a reduction in CST1A or an increase, for example, in Prevotella. But these are associations, and we really want to make the message careful and also said, we have to do intervention studies to validate these associations. And recently we got funding to do an intervention study. So, and again, we had, I think, almost 1,000 people who were willing to participate in this actually very tedious intervention because they have to wear 1, 2, 3, 5 different menstrual hygiene products. The same woman, they have a lot of menstrual cycles. They have to bring in all the samples, but they're willing to do it. And we recruited 100 women based on a bit of balanced groups. And so this is called the Luna project then as a kind of daughter project. And what is also very nice is that we got the funding to do it in Belgium, but also in Peru and Cameroon, so one of the countries in Africa. And that is something which I find one of the most rewarding aspects of the project. Is we didn't plan it, but because we had a lot of social media on the project, even before everything was published, is that local researchers across the world were inspired and asked, how can we do something similar? So they kind of organized it bottom up, and we just gave them advice or shared our experience, and they have been launching similar projects in their own home country. And every time they named the project after a strong female role model in their country. So the first early adopters were Laura in Peru, but also Cameroon and Nigeria are one of the early adopters. We also tried to get funding to do a similar project in South Africa. Unfortunately, it's not yet funded, so we don't give up. And yeah, for us, also for the similar ones, because it's always driven by local researchers, and we also always have to try to look for local funding. So to end my presentation, just some pictures from the project in Africa. This is a project actually initiated by Josiane Kenfak. She's a PhD student who found us online, and she really wanted to do something similar in her home country. It's affiliated to the University of Yaoundé, so the capital, but she also wanted to do the citizen science approach in a more rural area, you have to see how to pronounce Yunungo village, and she was able to motivate or to go there and actually to motivate the chefs of the village. So together with a translator, she convinced that she was allowed to do this citizen project there. So she convinced also the men that this was an important project. She was giving a lot of explanations. Here you see her standing with also the banner made for the project. And then I also put some pictures on the sampling campaign, because it was not easy. So she had to go with the motorcycle and the ice packs and go back to the lab. Then we tried that she could do all the sequencing and DNA extraction in her home lab, but it was not so easy. So then we arranged that she could come to our lab in Antwerp with the samples, which was again not easy with all the visa issues. But these are brand new results, I think, from 2 weeks ago, showing the data from the healthy women of Cameroon. And I'm super proud of her because she did also all the analysis herself. What I'm also really enthusiastic about is that in the rural area, for example, we do find quite a lot of Lactobacillus crispatus still, but also, of course, we also find a lot of inners and also more diverse ones. For example, Prevotella is really popping up, and I really want to understand more about what is it doing in healthy vagina. Is it really always a signature of more inflammatory conditions or bacterial vaginosis? And that's something, I think, for future research. And then another example I want to show is that also bottom-up organized by researchers in Kenya. They have named the project Dora in Nigeria to honor Dora, the previous Minister of Communication, also a role model fighting against, I think, misuse of fake medicines. And they organized a symposium a few months ago, also with policymakers and governmental people, and they also made their own infographics and also even translation to the local scientists. And I should also have shown it on the previous picture of Josiane, but also here it's nice to see that they make their own logo and design and t-shirts and communication. So, and with that, I would like to thank all the people involved, or especially all the sisterhood people. And this is a picture of the whole lab. 5 years ago, I had only one PhD student on the vaginal microbiome. It was really the smallest group in the team, but thanks to Isala, this has grown a lot. And finally, we can do more fundamental research on vaginal microbiome. With that, I would like to end. So I'm part of ICEP, and I was also asked to mention that they have grants and also awards for researchers from across the world, and you can apply if you're interested to participate. Thank you very much for listening.
Thank you so much, Sarah. As before, we'll take questions on the panel after the session. Our next speaker is Prof. Janneke van der Wijgert, who is a professor at the University Medical Center in Utrecht, the Netherlands. She's a clinician scientist and has performed clinical epidemiological research, lab-based research, and is very dedicated to product development to improve diagnosis, treatment, and prevention is a very important one. Thank you, of HIV and STIs. And what Janneke has spent a lot of her life working in Africa, particularly in Rwanda, South Africa, and Kenya, but other places too. And I think she's going to tell us a little bit more about her product development today. But we're very excited to have her. Thank you very much.
Thank you very much, Heather. I did it myself. Yes. So today I'm going to switch to a little bit more of a clinical story now. And in order to do so, I would like to give you my own interpretation of all of the data that's out there, because I think there has been a lot of descriptive work which has all been, game changing and really interesting, but not all of it is clinically relevant. I think we also really need to be able to differentiate between different types of vaginal microbiome compositions that require different types of management. So this is a slide that I made that is my own interpretation of the clinical relevance of all of the data that we've accumulated so far. So I think no dysbiosis, as people have already said. Women are either dominated by Lactobacillus crispatus or INES. Those are by far the most common two lactobacilli. And then there is a few other lactobacilli, but they are less common, or sometimes women do have a mixture of lactobacilli. I would call all of those like the absence of dysbiosis. And then occasionally, actually quite often, women will have lactobacilli, but they will also have very low levels of Gardnerella in particular, but they might also have very low levels of other anaerobes or low levels of yeasts like Candida yeast. And if those levels are low enough, I would also say no dysbiosis. But of course, the problem is that those organisms can start to grow, and then at some point it does become a clinical problem. And I think this is one of the open-ended questions: is where is that threshold? And then, of course, we've heard a lot about bacterial vaginosis, which is a clinical condition where anaerobic bacteria really dominate, and it's a mixture. It's always a mixture of anaerobic bacteria, very high diversity. And occasionally we also see women who are completely dominated by Gardnerella. Once in a while, I've also seen women dominated, for example, by Prevotella, but not very often. If we see one anaerobe dominated, it's often Gardnerella. So Gardnerella is an important one, I think. But this one, I think there is also consensus. This is bacterial vaginosis, and we would treat it with metronidazole or clindamycin. But I think there is also some other conditions that we might want to pay attention to, which I've called dysbiosis with pathobionts. And this is when women don't have not necessarily have high levels of anaerobic bacteria, but they have high levels of gram-positive pathobionts like streptococci. I think we all know, for example, that group B streptococcus is associated with preterm birth and with neonatal sepsis. And these are really fundamentally different types of bacteria from the anaerobes, and often also require a different type of antibiotic for treatment. So I tend to kind of look at them differently, from the bacterial vaginosis. And then there is also the gram-negative pathobionts, which are very much associated with urinary tract infections. And those bacteria we also often find, both in the urinary tract as well as the vagina. But what we see both in premenopausal and also in postmenopausal women is that the streptococci really seem to have a preference for the genital tract, and E. coli and other gram-negative bacteria really seem to have a preference for the urinary tract. And then, of course, there is Candida yeast. If women have high levels of Candida, that also requires a different type of treatment. That would have to be treated with antifungals. I think this is preaching to the choir. We already, we all know that dysbiosis, all the different kinds that I just mentioned, are really important in women. They can cause serious complications. So when you have dysbiosis in the reproductive tract, there is an association with HIV acquisition, HPV persistence, which can lead to cervical cancer, pelvic inflammatory disease, infertility, adverse pregnancy outcomes, and maternal and neonatal infections and sepsis. And all of these conditions tend to be more prevalent in Africa than they are elsewhere in the world. So this really is very important, particularly in the African setting. And then also in the urinary tract, which we, I think us vaginal researchers often overlook. But there is also some serious conditions there. Women can, particularly in postmenopause, many women develop recurrent UTIs that they just cannot get rid of. I'm currently doing a study on that. And it's, women are desperate. They're desperate. They just have constant urinary tract infections. The infection can go to the kidneys. They can develop urosepsis, which is a very common type of sepsis in older women. Kidney stones, incontinence, bladder cancer, all associated with microbes in the urinary tract. And these are also very, very common conditions that we actually know. We don't really know how to, for example, recurrent UTIs. We don't really know exactly how to treat them well and prevent them from happening. So the focus of my talk today is how can we now use all of this fantastic microbiome knowledge that we have accumulated in the last 20 to 30 years to improve things that are important from a clinical point of view. So how can we improve diagnosis, use the microbiome knowledge to improve diagnosis, but also to design novel treatments and prevention. I'm not going to talk about that today because there will be other talks about this at this symposium. And also to improve clinical care, screening, prevention, and surveillance. But most of what I will talk about today is about improving diagnosis, because diagnosis is at the root of all of these things. You first need to have a proper diagnosis before you can manage an infection well, or before you can do proper surveillance. So how can we use the microbiome knowledge to improve diagnosis? As most of the clinicians in the room know, at the moment, diagnosis is usually just based on symptoms, and this is also true in the Netherlands. I don't know about all of Europe, but definitely in the Netherlands, where the clinicians do a presumptive diagnosis. They do a clinical exam and they question the patient, and then based on that, they don't do any lab testing and they make a diagnosis. Here, as many of you know, often syndromic management is used. Women come in with vaginal discharge and they're treated for all the organisms that might have caused the vaginal discharge. We do have a lot of tests available for individual pathogens, but these are usually not assured. This is this WHO term that someone else, I think, mentioned yesterday. So many of these tests are actually expensive. They require equipment. They require a lab environment. So they're basically not available. And again, this is not just true in African settings. It's even true, for example, primary care physicians in the Netherlands. They don't have really good access to diagnostic testing. And then, of course, the other part I think that's really complicated is that the diseases that we talked about, so like things like bacterial vaginosis, urinary tract infections, they usually are they're multi-organism conditions. So they don't usually involve just one pathogen. They involve different types of organisms. Some of them are commensal, so they're always present and they only become a problem maybe when they go to the wrong place or when they multiply too much. So it's very difficult, actually. It's a very different type of diagnostic that you need compared to when you have a single pathogen. And then also some of the syndromes that we're talking about, like vaginal discharge syndrome, can have multiple causes. It can be a yeast, or it can be bacterial vaginosis. So this makes diagnosis even more complex than for other infections. And I've put this, I published this a few years ago. It's a slightly different from what Heather just presented, kind of integrating the different types of situations. So I think, when you have lactobacilli mostly, there tends to be, and we've, us and also other researchers have actually investigated this. Joanne, by measuring vaginal cytokines, we know that this doesn't really cause inflammation. I think we all believe that Lactobacillus crispatus is a bit better than inners because it's more stable and it creates a very healthy environment. But even the other lactobacilli, there is little inflammation present. But then when you develop anaerobic dysbiosis, which can be with or without a biofilm, which also would require a different type of treatment, maybe eventually in the future, or an STI, this definitely causes inflammation and inflammatory complications. But the same is true for candidiasis. And probably also, although this is less studied, when you have very high levels of Streptococcus, for example. But what I've shown here is, they're completely different. The vaginal environment is very different in these different types of situations. So with anaerobic dysbiosis, women tend to have a high vaginal pH, lower estrogen is often associated with it. Whereas when you have candidiasis, the pH might not be affected very much. So you would measure a healthy pH, so to speak. But if you then have a lot of yeast, then you still have a lot of inflammation. And you're still predisposed for acquiring HIV, for example, when you're exposed to HIV. So just measuring the vaginal pH would not be sufficient to pick up all of these different types of conditions. So what we did to try to, I was getting a little bit tired of, this is so complex and it's so difficult to do a good diagnosis. So we did a study where we kind of this was a study in Rwanda, where we used some of the rapid diagnostic tests that were already available at that time. And I knew that some of these tests were not assured. Like, for example, we used a gene expert for gonorrhea and chlamydia, and that's an expensive test and it requires equipment. But I thought we need to start somewhere. We should move away from just doing syndromic management and see if we can improve the algorithms by incorporating a few rapid tests that are already available. So we focused on vaginal discharge syndrome and lower abdominal pain. And we basically offered all of the women coming in with these conditions a rapid test with the TV-OSUM is for Trichomonas vaginalis, and also a vaginal pH swab. And then we did a risk score, and women who had high sexual risk, we also did a gene expert test for gonorrhea and chlamydia, because we knew in the Rwandan setting it would not be feasible to offer this to all women. But maybe after risk scoring, offering it to the women at highest risk, we might already improve on syndromic management. So we recruited 705 women who were sexually active and at risk of acquiring HIV or sexually transmitted infections. And we employed our own algorithms that had these rapid tests incorporated. And then also collected samples from all the women that we later on tested with gold standard molecular tests, so that we knew whether they had an infection or not. And we also collected symptom data so that we could recreate the syndromic algorithms. But the women were treated based on the WISH algorithm. So if they had a positive rapid test, obviously they were treated for that condition. And this is a lot of information, and I'm not going to go through it. This is the algorithm that we used. And I think the most important thing is what I just said. All the women received rapid tests for Trichomonas and a vaginal pH swab. Women at high risk also gene expert testing, and everyone, some of the other conditions were treated syndromically, like genital ulcers were treated syndromically. And this is some of our results. On this slide, I'm showing the percentage of women that ended up receiving treatments. the wish is what we really did. And then sometimes when tests came back by gold standard, then people were recalled to the clinic to also receive treatment. This is hypothetical. This is what would have happened if we had followed WHO procedure. And what you can see is that if we would have followed the WHO, this is obvious. This is the old algorithm because they've changed it now. But then more than half of all of the women would have received multiple antibiotics and antifungals. A lot of overtreatment. When we used our wish, the gold standard, this is the actual prevalences of the infections that we found. So it was quite a high risk population with at least 40% of the women had at least one infection. 8.5% chlamydia, 7% gonorrhea, 16% trichomonas, 18% BV, and 8.6% vulvovaginal candidiasis. And when you compare WISH to the gold standard, then you see that we did quite well with the chlamydia and the gonorrhea. We missed a few infections, but the sensitivity of the test of course is really good. And also the TV OSAM did quite well, but we actually still really overtreated the bacterial vaginosis, which was based on the vaginal pH. And this is basically the same information, but we looked at sensitivity and specificity. So again, you can see that it was actually quite good for most of the infections, except for the bacteria, for the BV and BV mostly. So so how far did we get with incorporating these point of care tests? So basically, I think it worked well for gonorrhea and chlamydia, but as I already told you, this is still an expensive test, so it's not something that can be rolled out on a large scale. TV also worked quite well. This is something that might be done on a larger scale, but for BV and candidiasis, we didn't really succeed. So we tried, a few other things to try to improve the algorithm. So we looked at different vaginal pH cutoffs to see if we could reduce overtreatment. We looked at, if we were to do a vaginal pH first and then add a confirmatory test, which can be microscopy, Nugent microscopy, but it could also be a molecular test. We could indeed increase sensitivity and specificity significantly, but it would require a lot of confirmatory tests. Remember, this is 700 women, and we would have had to do 275 confirmatory tests. We also try to improve the candidiasis performance and were to only treat women for candidiasis if everything else was negative. And we were able to improve the sensitivity a little bit, but it's not optimal, obviously. There were some other observations in this data set. We really, I'm sure many of you in the room have found the same thing. Symptoms and signs did not at all correlate with whether women had an infection. That was very clear in this data. And this is also signs. Signs is what the clinicians observed. That didn't correlate either. So it's not just what women tell you, but also what the clinicians observe during a pelvic exam. It doesn't correlate with the presence of an infection. We also try to restrict. We did these algorithms on all the women, whether they were symptomatic or asymptomatic. If we restricted it to the women who did have symptoms, it still didn't perform very well. We also observed that very few sexual partners were treated, which is bad. We need to do much better on partner treatments. The staff really liked working with the point of care tests. They liked them, and they were also highly acceptable to the clients, except of course waiting for 90 minutes for the gene expert. That was the least popular. So moving forward, luckily the WHO now in the new guidelines that came out in 2021, they are recommending doing diagnostic testing if available. So they have now basically added that to the guidelines, which I'm very happy about. I know there are many settings where diagnostic testing is not available, but there are settings where it is available. So, when it is available, go for it. And I think we should get started with the point of care test that we already have available, but obviously I've already pointed out there are many caveats, so I think we need new diagnostics. And for that, I think we can use all of the microbiome data that we have. Oh yeah, I added this slide just to make a point that the diagnostics do need to be designed well and validated, because I've also seen examples of tests that I think are actually unsafe for patients. This is one from my own country, from the Netherlands. This was a molecular test that was put on the market for women undergoing in vitro fertilization, and they can buy it online. And the physicians don't know how to interpret it. The women don't know. I get emails every day because people look me up online, and I have nothing to do with this test, by the way. And then they ask me, what should I do? It says that I have this and these bacteria, and what should I do? And so, yeah, it's ridiculous that a test like that is actually admitted to the market. But even the more benign, the companies that really mean well, the tendency now is to do a molecular test that will detect lots of different organisms. And then the physicians, they don't really like that. They just want to know the organisms that they know that they have to treat, the ones that are clinically relevant. They want to know, do I have to treat BV? Do I have to treat vulvovaginal candidiasis? Is there an STI present? And they don't want to know all of the other results because they also worry about liability. It's been detected, and if they then deliberately do not treat it, it opens them up for criticism and even for lawsuits. So a lot of the clinicians don't like to use these molecular tests that test for like 30 pathogens at the same time, or 30 bacteria at the same time. So here is some of my ideas on what we should be thinking about. I think we really need to think carefully about which targets to include in those types of tests. But not only that, I also really think that quantity matters. So as I mentioned before, if you just have a few vaginal yeasts, that probably isn't the end of the world. It only becomes a problem when the yeasts start multiplying and they start causing a lot of inflammation. And that might be before a person develops symptoms. And we don't really know where that threshold lies at the moment. So we need to figure out when do we really need to intervene? Often we really don't know. So we need to look at organism loads. As you all know, because Joanne is here at UCT, maybe we should also look at inflammation, quantify the inflammation. That might also give us important clues on what to treat. And also look at, define the clinical and the public health needs that we have. Is it just about improving quality of life, so that women can have their symptoms treated? Or like, for example, in high-risk women, we would like to prevent HIV. In pregnant women, we would like to prevent pregnancy complications. So we might need different diagnostic tests for these different groups. And of course, the tests need to be assured. We would like them to be widely available, and that, even in rural clinics, they can use them. And I just want to acknowledge a lot of people. Also, Tanya, who is in the room, she was involved in a lot of these studies. Her lab did a lot of the testing for the study that I just presented, and the team in Rwanda that conducted the study.
Thank you very much. Our next speaker and our last speaker for the session is Dr. Indriati Hood-Pishchany. And she's a clinician scientist who just started her lab at the University of North Carolina at Chapel Hill. And Indriati studies microbial ecology and the vaginal microbiome, integrating molecular microbiology and ecological principles to better understand how vaginal microbes interact with the host. And what's especially exciting is that Indriati is spending 2 days with us after the symposium, despite having a small toddler at home and a new lab, to teach us how to use these very exciting bioreactors, which she's going to tell you about today.
Thank you, Heather, for that kind introduction. And thank you to all of the organizers. It's been wonderful to be here and hear some exciting talks. And to share some of the work. So I started my lab about a year ago at UNC, call it the Carolina Vagina Lab, and we're really interested in microbial ecology and understanding how microbial communities interact, how they assemble, and in particular, understanding what bacteria eat. So how do they get their energy? What kind of micronutrients do they need? When I started my work in the vaginal microbiome, I think one of the things that I was thinking about is, what are some of the barriers, not only to therapeutic development, which I have up on this slide, but also to really just understanding these microbial communities. And I think I'm an infectious disease physician by training. And so when we think about infections and pathogens, we often think about single pathogen problems. So one microbe that causes a problem. But the vaginal microbiome and conditions like bacterial vaginosis are really fundamentally problems of microbial communities, and our tools and approaches and also our therapeutic development pipelines really aren't well equipped for studying microbial communities. It's also a problem affecting women, and I, of course, don't have to tell this audience that for a very long time, funding for problems that primarily or differently affect women have been largely underfunded. Thankfully, there's been movement to help that. But it's also a problem affecting vaginas, which I think is important because the human vagina is really quite unique. And so there's really just not any animal model that perfectly recapitulates both the physiology as well as the microbiology of the human vagina. And so this really leads to a lack of adequate preclinical models. And so the work that I'm going to show today is going to talk a little bit about more on sort of the microbial side and what we've been doing to try to understand the diverse microbes that are present in the vagina, and then touch a little bit on our work trying to develop tools to study microbial communities in reductionistic ways, but also to try to bridge that into preclinical models. So what is our general approach? We start with the bacteria, and really what we wanted to do is move beyond just a few species or a handful of strains, and really try to understand the sort of breadth and complexity of the vaginal microbes. And I will say that we have only really touched the surface of that, I think in particular because of the sort of origin and what we've had available to us to study. So we start with these individual microbes and try to understand their individual phenotypes. What do they grow on? How do they grow? We can then do comparative genomics and functional comparisons to try to understand the underlying basis for some of those phenotypes that we observe, and then really try to bring it back to the ecological context. How do those individual phenotypes affect the microbe's ability to actually compete within a community? And then also try to integrate with top-down methods. So what can we learn from the big metagenomic or metabolomic or other studies in people to generate hypotheses and test those then in reductionistic ways? So I'm going to talk a lot about glycogen today. And I thought I would start with a sort of brief history of what we know about glycogen in the vaginal microbiome, in particular because I think with a lot of things that we know about the vaginal microbiome, there's been a long history of observation. And as we start to dig down into the molecular details, we're starting to learn that some of what we know is maybe not what we thought it was. And so for decades, actually almost a century, we have sort of known that there's this association between vaginal glycogen, a glycogen produced by the vaginal epithelium, and lactobacillus colonization of the vagina. So studies looking at sort of the transition to puberty and this initiation of glycogen production, and the appearance of vaginal lactobacilli within the vagina, and then more recently, sort of quantifying the glycogen content and associating that with lactobacillus levels and low pH. And this really led to a model that's cited in many papers within the field that glycogen really supports lactobacillus dominance, that lactobacilli can ferment this carbohydrate source to produce lactate and lower the vaginal pH. Subsequent studies, beginning as early as the 1960s, that isolated lactobacilli, and really even as recently as 2008, found that many of the lactobacillus that were isolated from the vagina didn't actually grow on glycogen in vitro. And so this led to some refinement of the model in which human alpha amylase, which was detected in vaginal fluids, was thought to be the main driver of glycogen breakdown, and then subsequent utilization by lactobacilli. But still supporting this idea that it primarily supports lactobacillus growth. Fast forward about 5 or 6 years, and Van der Veer and Hertzberger and others showed that some lactobacillus isolates, in particular Lactobacillus crispatus, can in fact use glycogen, and they identified a putative glycogen degrading enzyme that sort of correlated with this phenotype. Working with Emily Balskis's group at Harvard, we characterized the biochemical nature of this enzyme and showed that this putative pololinase is in fact a bona fide glycogen degrading enzyme. So this tells us a lot about the relationship between lactobacillus and glycogen within the vagina, but what about all of the other bacteria? This is sort of where my work comes in. So as I mentioned in the beginning, we were really interested in studying the breadth of vaginal bacteria. And so we really started by just trying to amass a collection of microbes. Being in the US and being in a lab at the time that actually wasn't a vaginal microbiome lab, we started with what we had accessible, which was mostly isolates from the Human Microbiome Project, which are mainly North American isolates. We did eventually, through some collaborations with the Quan Lab, obtain some isolates from South Africa as well. But largely, this is a North American data set. We did basically high throughput bacterial cultivation to get growth characteristics from these different bacteria. What I'm showing you here is essentially bacteria arrayed vertically, carbohydrates arrayed horizontally, and the more red a bar is, the more that it's growing. And we're mostly just focusing on this column for today. These are different forms of glycogen or amylopectin, which is very structurally similar. And what you can see is there's a lot of red bars kind of throughout these columns. The Lactobacillus crispatus isolates mostly cluster here, L. inners mostly down here. But then we also see there's a lot of red within Gardnerella, within Prevotella, and then even among some of these sort of other diverse anaerobes. And this is not totally surprising. In our study with Emily's lab, we actually did characterize glycogen-degrading enzymes from Prevotella, Gardnerella, Lactobacillus inners, and Mobiluncus. Larry Forney's group had shown that they could detect bacterial amylases in vaginal fluid using proteomics. And Jacques Ravel's group had also shown that predicted glycogen-degrading enzymes were highly expressed across different CSC types, here 1, 3, and 4, and importantly, that these were expressed by many different types of bacteria. So what this all tells us is that glycogen degradation is actually a very common feature among vaginal microbes and in the vaginal microbiome. And so how does glycogen utilization actually impact communities? So to get at this question, we started by looking at synthetic communities. So we assembled these 5-member communities that had bacteria that are commonly found with each other in the vaginal environment. And we did experiments where we would grow them on different carbohydrate sources and swapping in and out a Lactobacillus crispatus that either has a glycogen debranching enzyme or does not have it. So I'm showing just one example of that here. So here's the relative abundance on the y-axis and the sugar that they're growing on the x-axis, and the different colors represent different species. And so if we start first with the Lactobacillus crispatus that lacks the glycogen degrading enzyme, so it can't grow on glycogen on its own. What we see is that during growth in maltose, the lactobacillus here in this lighter blue color is actually growing quite well. But when we grow it in glycogen, it actually is almost completely eliminated from the community and replaced by Lactobacillus inners, which is a glycogen utilizer, indicated here by the little Pac-Man. If we contrast that with a Lactobacillus crispatus that has a glycogen degrading enzyme, what we see is that similarly, it grows well in maltose, and then it competes a little bit better with Lactobacillus inners when it's grown in glycogen, telling us that L. crispatus isolates that degrade glycogen seem to have an advantage during growth in community. What we found is that we can actually rescue the growth of the non-glycogen utilizer by just providing an exogenous enzyme. So this essentially is returning the community to something more like what we see in maltose. And it even provides a little bit of a growth advantage to the strain that even has the glycogen degrading enzyme. And then this is just showing this another way. We're looking at the absolute abundance of the 2 L. crispatus isolates in their respective communities, and we see that they're similar in maltose, that the one that can't degrade glycogen does poorly in glycogen, but if we supplement with an enzyme, we can rescue its growth. So when we did the experiments with Emily's group, one of the things that they found in comparing these enzymes is that they actually had different biochemical properties and to some extent some differences in the sort of ratio of breakdown products. And so one of the questions that we asked is whether only an L. crispatus enzyme could rescue L. crispatus within a community or whether other enzymes could also rescue. And what we found is that that actually it doesn't really matter, that any enzyme that breaks down glycogen is able to rescue. So this is, again, looking at the absolute abundance data of just the L. crispatus within this community. grown with a Gardnerella that lacks the enzyme, but then providing an exogenous Gardnerella enzyme. And then again, we see this rescue and growth. What was particularly interesting, though, is that if we actually swapped out this Gardnerella with one that produces its own enzyme, we found that that actually also rescues L. crispatus growth, suggesting that when you're growing the Gardnerella that can degrade glycogen, it actually produces public goods that are available to other bacteria to use, and that supplying an exogenous enzyme didn't really further increase that. And what was particularly interesting is that if we look at the total bacterial abundance within the entire community, it actually increases in the presence of this Gardnerella, suggesting that this Gardnerella is actually increasing the total carrying capacity or the ability of the whole community to access this nutrient source. And you might wonder, okay, well, this is in vitro, does that really matter? And it turns out that a number of studies have sort of indicated that during bacterial vaginosis, bacterial loads may be somewhat higher. So just one example from Heather's group is that in their study looking at copper IUDs in association with BV, they found, looking at 16S copies, that there was a significant increase in total bacterial load in BV conditions. And so our data might suggest that one of the mechanisms for that may be by sort of better accessing the nutrient source within the vagina. So why is it that the Gardnerella is able to do this and we don't see the same effect with lactobacillus? Well, one of the reasons we think is that the Gardnerella enzyme turns out to have a very high activity compared to other enzymes that we studied, about 10 times higher processivity for glycogen. But one of the other things that we've recently found is that L. crispatus also seems to be a little greedy when it degrades glycogen. So when we grew each of these three species individually on glycogen, each of these are glycogen utilizers, we found that while Gardnerella and Elinors had some sort of sloppy degradation and left some maltose, had some maltose left over, L. crispatus, no matter what time point we look at, there's just no maltose there. It's just all gone. And we know from the enzymatic data that they produce maltose. Their enzymes certainly produce it. And so it seems like, and we're trying to better characterize this molecularly, that they take it up very quickly from the environment. So just to kind of summarize what we've talked about today, so we know now that glycogen-degrading enzymes are produced by not just lactobacilli, but many other species in the vagina, that this leads to the release of breakdown products. And in the case of Lactobacillus crispatus, dominant communities on the right, they may take up that maltose quickly to produce lactate and lower the vaginal pH. And data we haven't shown, and that many of people have shown, that this has both direct effects on potentially inhibiting enzyme function, but also on sort of more broadly on inhibiting bacterial growth. And then what we've also learned is that the Gardnerella enzyme seems to be this very high processivity enzyme that really just quickly degrades glycogen. And what we'd like to suggest is that potentially the association between high glycogen and lactobacillus is not so much that it's preferentially supporting lactobacillus growth, although certainly lactobacillus degraders seem to have, glycogen degraders seem to have an advantage, that maybe what we're seeing is actually that when Gardnerella or some of these other bacteria are present, they're actually just degrading and utilizing that source much more quickly. So I'm going to just very briefly touch on where our lab is going. So we're really interested in dynamic models for studying microbial communities. If you want to study bacterial state transition, so going from a CST4 to a CST1 or vice versa, you really need a system in which you can dynamically study those transitions. And so beginning in my postdoc lab, working with a system called Evolver, and now in my current lab, working with the system bioreactor, we've adapted this small bioreactor system to be able to study microbial communities in a way that we can actually dynamically sort of achieve a steady state, introduce perturbations, and measure response to those perturbations. So just kind of briefly giving some examples of that, here we've grown a BV-like community. We see that it takes a few days to achieve a steady state, and then without further perturbation, it remains relatively stable. Importantly, if you look at these low abundance members, we find that they remain, they persist even out to late time points. And this is important because if we're getting extinction and they're just going away, we're not going to be able to perturb the system and have them come back. And so just giving an example of our ability to sort of perturb, here we've grown two different communities and introduced lactobacillus. In one community, the lactobacillus takes over and is able to engraft. In the other, it's really unsuccessful. We see a couple of points here. One is that when lactobacillus is dominant, we again see that lower total bacterial load, whereas in this mixed community, we see a higher bacterial load. And then importantly, we introduced another perturbation here, which I wasn't going to get into in detail. We were trying to mimic menses by adding blood. And we do see this outgrowth when we make those perturbations. And although we're not seeing a huge shift in the total bacterial composition, the fact that we can get some of these lower abundance members to sort of outgrow at these different times suggests that it is a dynamic system in which we can introduce these perturbations. With that, I'll just thank the members of my lab, people who were brave and took a chance in joining a brand new lab where they had to open a lot of boxes. My former lab, Seth Ratkoff-Nahum and his group at Boston Children's, the Bauskas lab, the FRESH study participants, and the Vaginal Microbiome Consortium, all of which have contributed some isolates to these studies.
Thank you so much. Can the speakers please come and sit in the front? And I've been asked to ask people to try and squish a little bit in, because it's really hard for the people with microphones to get to people in the middle when there's this very peripheral seating pattern, if that's okay. Thank you. All right. All right, so thanks very much. We have, please, we can take questions. I think there's one from Anna over here. And maybe just, I know I'm not very good at this, but introduce yourself before you ask your question.
Sure, I'm Anna Happel from the University of Cape Town, and I have a question for Sarah. Really inspiring talk. I still remember meeting your PhD student, when Isala just started, and it's amazing to see how far it has come in the last few years. I was just wondering whether you could comment on lessons learned with regards to science communication from, the Belgium project, and maybe also from sisterhood projects, because yesterday we talked about how important it is to, involve participants more, and that one of the way to empower women would be to give back results, which you have done in the study. But then, Heather raised some concerns, and I think it is a valid concern on how to communicate science in a way that, educates women without scaring them. And I think you touched briefly on it, but I just wondered if you could expand on it.
Thank you very much, Anna. Is it on? There are many lessons we learned. I think the first lesson we learned was that when I was planning this project as a basic scientist, so I'm not a medical doctor, many people were telling me, oh, this is a taboo topic. It will be very difficult to motivate women to talk about it. And that was not the case. And it doesn't, even also now in a lot of sisterhood countries where you think of vaginal health or even sexual health, or women taking their health into their own hands, it might be a taboo. It doesn't seem to be like that. So it's if you find a narrative that women feel comfortable to talk about this and to participate to studies, it can actually motivate a lot of women. But it's also true, you have to be really careful with your communication. And now in this area that everything has to be short, and it's not always easy to balance it out. So what we did a lot was we had a whole advisory board of medical doctors who are, of course, crucial. Go to gynecologists and the GPs. I think that's also a very good balance, because they have different viewpoints, because they see different patients. So it's good to have this balance. Then we also had social scientists and communication experts. And that's also a lesson we say to all the sisterhoods: make sure that you communicate first, or you do tryouts with your advisory board, with communication specialists, with other social scientists, so that you are careful with that. Then another thing we learned is that the sisterhoods, or if they want to do their own project, you really have to adapt to the local language and the local habits and the local communication style. So do your proper homework first before you just launch a call. And then I think indeed the tricky part is also that science is not ready to tell you, okay, you have probably this disease, or this is bad, or this is good. But often women they often ask us, give us a tip, what should I do to have a better vaginal microbiome? And then for me, say, we only know associations. There are a few things we can say, but we need to do more intervention studies to be able to give you new treatment or suggestions.
Thanks. I'm wondering, if Janneke, if you want to add anything from your experience?
Yeah, I think my experience has been very different from Isala, because in Isala you really thought about the communication and you had people there, communicating, and it was all well prepared. Whereas the example that I mentioned was a test that was put on the market without anyone thinking this through. So they just made it available, and people started ordering it online and sending the sample into the lab and receiving results, and they had no idea what to do with the results and panicky. And these are people, undergoing IVF, which is very expensive, and so there was. So I really felt that was an example of how it should not be done. And what I was really surprised by when I first started getting emails from people is that people are allowed to put tests like that on the market. There is no regulation, which is strange, because when you work on interventions, we all know you have to go through a very extensive regulation process and medical ethics process. And apparently with diagnostics, it's very easy.
Thanks. Tony?
Good morning. Tony here from the NGS Academy team. I'm learning a lot. Thank you to all the speakers. So a lot of the times your case studies or your research is really dictated by what type of sample and how much, or the quality of the sample that you can ascertain. And for us, we're doing a training with Africa CDC on monkeypox in literally a week's time. And we have participants from 16 different countries and institutions coming. And we're really, really struggling to get clinical data, patient data. I know in Europe there's GDPR, in South Africa there's the Puppy Act, and many of these African countries also have, personal protection that we just can't get. There's too much red tape. And I was really flabbergasted to see how much participant you're able to, get for your research. Can you perhaps just comment, are you paying these participants? Is there any way for us to try to, get people on board? Because sometimes it's really hard. There's culture and language barriers. We can't get the people in the village to trust us and, the healthcare providers. And in a lot of the African communities, there's this legacy or distrust between the people and the government. And you just can't overcome that readily. Thank you.
Thank you very much for this question. I think first thing, we didn't pay the participants, and we also did a feedback survey afterwards, is that the reward of participating, women felt more that let's write history together, let's advance science together. So the feedback was that they just wanted to contribute to science and felt heard. And then that was for the Belgian case. I know Josiane in Cameroon, for example. I fully agree. You have to really think properly on how you will organize your sampling campaign. So you have to go to talk to the villages. You need to have trust persons. Community healthcare workers are often very important to help you with the sampling. So for some projects, it takes 1 or 2 years before real sampling is started. But then, if you find a narrative and also explain that is, in our case, it was not to detect disease or not to pinpoint problems. It was more to advance science, that they could motivate a lot of women to participate. So it depends a bit. I think often healthcare workers, of course, if you go for HIV screening or HPV monitoring, then it's a bit of a difficult message. While now we had a message, let's jointly together, let's advance science, and we need to know more about this very important organ for human reproduction, and we need more research first. And that's a message that apparently is really motivating women across the world now to participate in studies like that.
And then in my experience with the Rinda Ubuzima team, the study that I presented, they have community advisory boards or groups that they work with. And they also work a lot with peers. So, for example, when we work with sex workers, we actually work with sex workers. They become part of the team. They give advice on how best, and they, so they are very trusted in their own communities. So they then go to the communities and explain. We also sometimes there are rumors that people are worried about certain things. Like, for example, if we have to take blood samples, people don't, sometimes they worry about that. And what we've done is actually invite groups of people from the community to come over to the clinic and to the lab to see exactly what we do with the blood samples. And we have educational sessions where we teach people how the body works, and that when you take blood, it doesn't mean that it's gone forever, but you make new blood all the time. This is something that a lot of people don't know about. So there is a lot that you can do to earn the trust of communities. You really have to earn the trust.
Yeah, I think that's really important. I think I want to add to that is that also as scientists, you need to be honest about what you know and what you don't know, and don't always be like, we know everything. No, you have to motivate the community to share their knowledge, and also acknowledge that they probably know also a lot of things we as scientists are not aware of. So make them more team members and give them also the possibility to co-create your research, and then the trust building goes much easier in our experience. Because it's true, as scientists, we don't know everything. We have our...
It's a lot of work.
Yeah, it's a lot of work. We need to spend time on that.
Okay, Prof. Bukuse.
Thank you very much, Elizabeth Bukuse, Cambrian, the University of Washington. Janneke, my question goes to you. I'm sort of a little disheartened to see that women in your country are also struggling with the same things. We think that women in sub-Saharan Africa are the ones who are disempowered, struggling with how to notify partners, how to come in for treatment, how to get partners treated. But when you see the same thing in Europe, then I wonder, what's the challenge? I mean, how do we overcome this?
Yeah.
If education, empowerment, having a better socioeconomic status, better status of living doesn't solve this, how are we going to resolve this?
Yeah, I think what I see in the Netherlands a lot is that the things that we do, the vaginal infections, urinary tract infection, postmenopausal women, it's just because the men have dominated the infectious disease internal medicine field for so long. They just aren't interested. Even now, when I talk to my male colleagues, they just aren't interested. They say, you do that work, that's fine, but they don't engage. They really don't engage. How to change that? I think the only way is to have more female scientists. We have a lot of female clinicians, but we don't, we still are lagging behind with female professors who have their own research groups like myself. When I started out as a student, I think there was about 5% of all the professors were female, and now we're at about 20%. So it's improving. And it really is the women themselves who are going to have to make that change, I think, when it comes to research, and then the research will be translated into different policies, and and it's happening, but it's happening slowly.
Caroline?
Hi, Caroline Mitchell from Mass General in Boston. My question is for Indriati. And as a clinician who desperately wants a model system, I want to ask your system to do everything, but which I know is not possible. I guess I'm wondering, how do you incorporate host features that Isala has shown us are important? How do you think about, you've said it's a reductionist model, which I think is exactly what we need, but how do you start to build on that? What other data do you need to make it a more comprehensive model?
Yeah, it's a good question. I mean, I think right now we're mostly relying on data that we see clinically. Sometimes that's still a lot of assumptions. So we see transitions during menses, and so we can think about ways that we can try to model that. There are some studies out there that look at, for example, proteomics of menstrual fluid, and we can start to take components of that and see, what actually changes the community. And then some of it is also a little bit around media design, and ways that we can try to incorporate something that makes it a little bit more of a human-like environment. I think there are other models like the vagina on chip where you have human cells there and it can get that sort of, the responses, which I think is important. I think that's something we can't model in this system. But as far as the more reductionist piece, I think it's trying to learn as much as we can from the human environment and from the studies, metabolomics and other things about what's there, but also recognizing that what we're measuring is, and I think Jacques talks about this all the time, has been already acted upon by the microbes. And so having a little bit of being a little humble, I guess, in what we're putting in, and knowing that, like, okay, we might be modeling something that's already been affected by the microbes and others that are there.
Can I ask a quick additional question, and then we'll get to you, Tariq? Have you ever, Janneke brought up something about Candida, and I think we've seen that Candida and lactobacilli can coexist. Are you able to add Candida to the system and see what the dynamics?
We haven't tried yet, but there's not necessarily any reason that we couldn't. It's something that's been on the list of things to do. Yeah, I think, it comes up all the time, and it's something that I've been really interested in trying to incorporate.
Okay, let us know. Okay, Tariq, and that, I think, is our last question.
Thank you. Hi, everyone. Thanks for the awesome session. A question particularly for you, Indriati. Is that
louder?
Thank you. A question particularly for you, Indriati. Yesterday, Professor Kwan spoke quite a bit about sort of what's in the vaginal microbiome, but then also the strain differences and how they interact with each other. And then today, you also spoke about kind of knowing what's there, and then showed really awesome detailed data about, what they're doing and how that impacts or how that may inform vaginal health and so on. So what is this what, in your opinion, would be the relationship between fully understanding what's there compared to what's there is doing, kind of thing?
Yeah, it's a good question. I think when I started my work, one of the first things that we noticed was the strain level differences in phenotypes across different bacteria. So among L. crispatus, there's a lot of variation in what they can do. And often you can find directly, if you look at the genomics, okay, we can predict, we could have predicted basically what they would have been able to do. Sometimes it's not as easy. Sometimes it's actually, there's minor point mutations or things that we find that are probably affecting expression. And maybe that represents a strain that's undergoing evolution already in the environment to sort of lose particular functions. And so I think understanding what's there and a sort of detailed understanding of not just 16S level sort of taxonomy, but really at a even predicted functional level through metagenomics, and that getting that strain level resolution, I think, can help to predict some of the functional consequences. I think we still need a lot of sort of reductionistic studies to understand what those functional consequences are. But I think, again, one of the things that was just a huge takeaway for me is that there is a lot of strain level variation. And so just understanding a 16S sort of profile doesn't really tell you a lot about what that community can actually do.
All right. Well, thanks very much to our speakers. They're great talks, and let's go get some tea.
Hi, welcome to the Celtic Molecular Diagnostics page. Join me as I tell you about who we are and introduce you to our product portfolio. Established in 2003, Celtic has a reputable history. With over 2 decades in business, we've thrived in molecular diagnostics and research. Our expertise also spans lab instrumentation distribution. Celtic supplies state-of-the-art products from over 25 suppliers. Our vision is to become the leading supplier of molecular biology reagents and products in Africa, and our mission is to give our molecular biology customers unmatched and superior service accompanied by honesty, integrity, and sincerity. Our brands cover a wide range of product categories, including products for conventional and real-time PCR, cell culture, nucleic acid extraction, as well as reagents for Western blotting, flow cytometry, and ELISA. Our study brands accommodate your entire workflow, from sample preparation to analysis. We also have a molecular diagnostic division, as well as diverse instruments in our portfolio. For inquiries into any of our products, use the contact details shown here to get in touch with us. And if you are ever in Cape Town, feel free to visit our offices in Kenilworth.